Dear students, in the previous video I talked about the NGS and how it is different from the other methods and how it is useful. Now we will talk about the steps involved in the NGS. So it is a library preparation, cluster generation, sequencing and data analysis. So the first one and very important one is the library preparation. So you look into this diagram how it happens here. So this is the fragment. So this is the DNA sequence which you want to sequence. What you did the fragmentation of this sequence and it is fragmentized, and then you add some adapters. So here it is a black color, I will show you in a diagram. Suppose this is a sequence and these are the adapters like that. So what is the need of adapters? Because when you will load this library into a flow cell, so with this sequence you cannot ligate it, because you don't know what suppose you just randomly break down, you don't know what is the nucleotide present here and what is the nucleotide present here, but we know about the adapters. What is there? We know this and this is known. This one is known, but this is unknown. So we add the adapters here, and when we load this one into the flow cell, so it can easily stick over there. If it is not adapters will not added, it cannot bind with the flow cell. So this is the first step of the library preparation. It is the fragmentation and the sequencing library. You can read it over here. The sequencing library is prepared by random fragmentation of the DNA or cDNA sample, followed by 5' and 3' adapter ligation. Alternatively, tegmentation, adaptation is a tegmentation, combines the fragmentation and ligation reaction into a single step that greatly increases the efficiency of the library preparation process. Adapter ligates fragment are then PCR amplified and gel purified. These are the process took place. Next is a cluster amplification. So what is the cluster amplification? For cluster generation, the library is loaded into a flow cell. You can see over here, this is a flow cell. What library you have prepared before, you just loaded over here. Now the method is saying cluster amplification, amplifying. When you are amplifying anything, the number of the things will automatically increase. So you can see over here, it is a bridge amplification process. Bridge means you will move between one place. So this is 1114 sequence where you can see, and here you can see it is a so many number of sequences are available. Okay, so this is the bridge amplification, cluster amplification. Library is loaded and the fragments are hybridized to the flow cell surface, and you will get the lots of sequences from there. Third one is the sequencing. Methods that detect single bases as they are incorporated into DNA template strands, as all 4 reversible terminator bound DNTPs are present during each sequence cycle. Natural competition minimize incorporation bias and greatly reduce raw error rates. The result is highly accurate base by base sequencing that virtually eliminates sequence context specific errors even within repetitive sequence regions and homopolymers. These are the sequencing method. Then we go for the data analysis. During data analysis and alignment, the newly identified sequence reads are aligned to a reference genome following alignment. Many variations, like there are so many methods are available for data analysis. Like this is the amplification you have performed. Then sequencing, as I told you before, the same methods occur, like DNTPs were added. It will just amplify either it is A, T, C, G one by one, and once this whole process will over, it will be, this is the read, and you have a reference genome. As we talked in the previous videos, you can go into the previous videos what I have prepared for the genome sequence annotation. You can go and understand how this happens. So this is your raw sequence and this is your known sequence. You will perform the alignment between these two, and you will find out and this is, you will find out the alignment in data analysis and how the whole process will occur. So, this is the overall process of next generation sequencing. Thank you.